All attendees are in listen-only mode.
Hi, everybody. Thanks for coming to the part 2 of our risk assessment and risk, basically risk assessment and root cause analysis webinar. Some of you have asked a couple of things. So one is you've asked about the first part of the webinar, and that has been posted. We had a little bit of technical glitch, but that's now taken care of. So you can all access the first part of the risk assessment and mitigation, as well as our introduction webinar. And this is just a reminder that tomorrow at 1, we will be doing the first of our eClinical Technologies webinars. As I put that together, there was so much material that we'll probably add another webinar at the end to talk about the specific technology areas, but this has a lot of information that will help you as far as your monitoring and how technology and what you need to do based on regulations and how you can use that for your monitoring activities. So let's get started. What I want to do is to just go through where we were and pick up from there. And also, don't hesitate to email me if you have questions. Somebody had trouble downloading one of the documents, so I was able to send all the documents to her. Also, if you have any requests or suggestions, please let us know. If you can like us on Facebook, if you can subscribe to our YouTube channel and Google+, those are a real big help for us, and that's how we'll communicate whether we've got new webinars or other information so that you know. And also, please let your friends know if you think this is useful. Although it's a series of 7, you can hop in at any time. So because we have all the previous webinars recorded, people can catch up, and we'd really like to train as many people as we can. So please share if you like it. Share it with your colleagues, and so that we can get a bigger community of people who are knowledgeable about risk-based monitoring. So let's get started. Where we were last week is we had gone through identifying potential risks in critical areas. So we talked about subject safety, protocol compliance, investigational product, GCP, and efficacy measures. So we talked about that this needed to be done at the very beginning, even before the protocol was complete, and that you do a second round after the protocol is complete to determine what are the areas that are going to take more significant oversight, and what are you going to be spending your time on now that you're not doing SDV. So this risk identification is such an important step, and as I said, there are tools out there, but you can use anything. Then you want to quantify the risk. Again, what this is helping you do is do your prioritization. So you want to figure out what's the likelihood, what would be the impact if it happened, what are the controls in place, and then you can think about how will you identify those risks, who will do it, and what will be the frequency. So you're going to quantify the risks, and then you're going to ensure that the data and reports are available and define who will review and the frequency. So we're through all of those. And now what we're going to do is talk about determining tolerance levels for risk. And the one point I want to make is sometimes that tolerance level is one case. So this is probably one of the harder areas, especially for smaller studies, but we'll show you some information about that. So let's move forward with determining tolerance levels. So one of the things that you want to do is think about things. So one of the places that you see a lot of deviations, everybody does, is in timing and assessment windows. So when is it critical? When are those windows critical? And when can you allow variability? So this also goes back to your protocol design. But, for instance, if you were doing a PK analysis, pharmacokinetic analysis, then a specific time point may be critical, where if you're going to do population pharmacokinetics and you just need to know when a subject took a dose of medication, when for that day, that's a very different thing. So part of it is to think about which are the windows that I really need to look at that are really, really important. For instance, dosing. Did they get dosed at the time that they were supposed to? Did they get the safety assessments at the time that they were supposed to? And then critical activities that may trigger additional activities. So for instance, I did this was several years ago, a program with a company at DIA, and one of their risk indicators they used was time to data entry. So remember, as our recommendation, as much as possible, is to have the sites enter the data contemporaneously with seeing the subject. But if you don't have that, then you do want to look at things like how long it took from time to data entry. And remember, this isn't just about making sure that you have the data to SDV. This is really about subject safety. So if we get that data right away, then we can begin to look at that, identify risk, and see whether the site is doing what they're supposed to. Clearly, a delay in SAE reporting, this may be, let's say they didn't report an SAE, this may be a tolerance level of 1 that you're going to say, or depending on your area, if you're doing a oncology study, then maybe missing one SAE in delaying that reporting may be not as critical. So again, all of these things really vary depending on your study. Critical activities that may trigger additional ones would be, like we talked about, too little variability in something like your blood pressure measurements across time and subjects, because automatically your antenna should go up and say, we need to look at this and see whether there's some evidence of fraud. So these risk thresholds are important, because what they're doing is saying, this is at this point when this site meets these risk thresholds, we need to do something. So the risk threshold is the amount of risk that will be tolerated versus something that must be further investigated. So, there's always errors. You're going to see those kinds of errors. Not all errors are the same, are created equally, just like not all data is created equally. So you want to think about, what can we be comfortable with, and at what point do we need to do more? Okay. And the threshold can be determined based on how far from the mean something falls. So I know you're going, ugh, I don't like statistics, but this is a very, very simple statistic, and is something that is important. So if you want to say that, and so we use what are called z-scores for that. So I think we were just finishing this poll when we did the last study. So in this example, this was the example where the screen failure rate is 20%. And so what are your thresholds that you're going to use, that you're going to say, okay, if they are within 5%, it's okay, but if they're out 20%, then that's not okay. So those are the kinds of things that you want to think about when you're determining your risk threshold. So again, that threshold can be an absolute number. So in that example, it could be something like 10, which I wouldn't agree with in this, because remember, one of the really important things here is that a lot of this data needs to be put into the context of how many subjects have actually been enrolled. So sometimes an absolute number is the case. Sometimes a percentage is a better option. Sometimes a z-score, which I'm going to show you now. Again, for smaller studies, it may be important to define thresholds based on an absolute number, because you may not have enough subjects at each site. But in larger studies, what we recommend is using something such as a z-score. And what this is the number of standard deviations either above, which is a z-score that's a plus number, or below, which is a negative. So let's look at this bell-shaped curve. Everybody's seen this before. And remember, so if you are one standard deviation above and below, that constitutes 68% of the population, if you will. If you go out 2 standard deviations, you're getting 95% of the population. And again, what you want to do here is to, you're thinking of I want to look at outliers. And you do need to have normal data, but you can assume, that's why I said you need to have a larger number of sites. But if you use a z-score, which is easy to calculate, you can calculate it in Excel. If you say, I'm going to look at z-scores above 2 and below 2, then you're going to only be looking at the 5% of the outliers. And what you may say is that this is the group I'm going to look some at, and then I'm going to look at even more critical ones that are above or below 3. So a z-score of 2.5 would be here, okay? So you could say this would be something that would be worrisome because they're already 2.5%, you're in that 2.5% that's above the mean, more than 2 standard deviations above the mean. If you had a score of a -1, I wouldn't worry about it at all. If you have a score of a -3, it depends. Sometimes a minus is important. Excuse me, my computer just jumped. Sometimes a score of a -3 is important. Sometimes a score of a +3 is the important, or sometimes it's the ones on either side. And I believe I have a poll here. Let me go to the polls. Let me just see. Let me look at the next one. Yes, let's look at, I think I have a poll here so that you guys can't fall asleep. So let me launch this poll. When would a z-score of 2.5 be a good thing? So we've been talking about tolerance levels, but sometimes it's actually a good thing. So your options are, okay, that it's time to data entry. Is if you are 2.5 standard deviations above the mean, is that a good thing? Is it number of AEs? 2.5 standard deviations above the mean, is that a good thing? Number of screen failures, 2.5 standard deviations above the mean, is that a good thing? Or is it the number of subjects randomized? Is 2.5 standard deviations a good thing? So we have about 40% of you have voted, so I'll give you just a couple more minutes to see if you want to vote, and then we'll close the poll. Remember, I don't know who you are, what you voted. This is really so that you get involved and begin to try to think of this rather than just let me tell you. All right, I'm going to close the poll, and here's our results. So time to data entry, 29% said that, and number of subjects randomized, 65%. Well, the answer is really 65%. And I think that you might have gotten confused on time to data entry. If the number had been minus 2.5, that would have meant that you are 2.5 standard deviations below the mean, which means that you are probably one of the fastest ones entering data. So in this situation, a minus 2.5 would be a good thing, but number of subjects randomized is a good thing. So 2.5 standard deviations is a good thing. That says that this subject is randomizing a lot of subjects. So again, it has to be looked at in context, but that's those are the results that you have there. Okay, now let's move on. So now what we want to do is monitor for occurrence. Okay, so let's figure out, how are we going to figure out if those things happen? Because that's the really important thing. So one of the things that we have done, and so again, as I told you, what we're going to talk about, our methodology, mainly because I can give you concrete examples, is because review is a cross-functional activity, and we are absolutely convinced that we do need that cross-functional review because people think about data differently. So the DM review, so we use as our overarching kind of document a data and document review guidelines. And that defines, again, it's tied back into the risk. So we start with those risks. We then go to, okay, how are we going to look at them? Is it going to be something in the EDC? Is this going to be a report? And then we talk about who's going to look at it and how frequently they're going to look at it. And then while each group does their job, then that information then comes back into the team discussion, which happens monthly. And that's just an example. Again, depending on your study, it could, if it was a very slow enrolling study, it could be something like quarterly, but usually monthly seems about right. So that doesn't mean the DM is only doing the review monthly. The data managers are doing their review um, in real time. So we start our review as soon as the subjects enter data. The site monitor the same way. Medical monitor reviews also need to play a role in that. And then we do our central monitor review. So this is the way. And what we've done is people say, well, why don't you put your review in the monitoring plan or the data management plan? But because we use a cross-functional review, we want the guiding document for a study to be this data and document review guideline, which is available to everybody. And everybody participates in the setup of this so that this is owned across all the functional areas. Now, one of the other ways that so let me just give you another example. So this was a high risk. Ratings are often high risk. So this was a study where we were looking at ratings for herpes labialis, so cold sores. Okay, and the natural history of cold sores is that they don't ever progress from 5 to 4. So they may go from 1 or 2 down, but they never go from 4 down. So this was where the subjects had to have their ratings done every day, regardless of whether it was Saturday or Sunday or whatever. And then what we did, because the rater performance was the key efficacy endpoint, is that we reviewed these ratings every day to see whether we were starting to see any that did not make sense, because we wanted to focus on those right away. So in this example, this subject here has a rating that looks problematic. Okay, so these all go, as we said, they may stay the same for several days, but they all go up. Okay, but this one didn't. So this tells us that this is a rating that we need. This subject's rating is something we need to be concerned with, and that is at subject 05. So the bottom line is, it could be any number of things. It could be a report like this, or a data visualization. It could be, I'm going to look at the outliers for things like those that don't report SAEs fast enough or don't enter their data fast enough. There's all different kinds of ways that you're going to look at your high risk data elements. And this is just to give you an example of one where, when we instituted this, this sponsor had lost an entire site from a previous study because the ratings had errors in the ratings. They weren't consistent. And so the site had to be, and 75 subjects or I think it was 75 subjects were eliminated because the ratings were so poor. So this is something that needed to be focused on immediately, and that's the way we did it. Now, I grouped these together because what I want to do, because they go together. So as part of the quality management, there is, you identify your risk, you monitor for the occurrence of those high risk areas, and then for those high risk areas where you find errors, you need to do a root cause analysis. So it's important to realize that you need to understand why it happened, and then you need to develop remediation based on the specific root cause, and then you need to monitor for correction of behavior. So all of these go together, and we see this primarily as the site monitor's job, because they're the ones in most situations. So if these are errors that are at the site, then we see this as the site monitor's responsibility to really understand why those errors occurred, figure out the best remediation, and then monitor for correction of behavior. Now, if it is something that has to do, let's say, with maybe because this could be that there's a question that is unclear that's in the EDC, so you're seeing a whole lot of queries across all the sites, then that would not go to the monitor. That would be something that would be managed. The root cause analysis would be managed by the data manager. If the issue was that the informed consents from a central study were in error, and I see this so often, then that, because that is a global activity, then that would go probably to the project manager. So it depends on what the issue is that you have as to who will be responsible. But if it's a site issue, we think these should go to the site monitor. Now, the reason I bring this up as being so important is this is something that we're seeing in FDA warning letters. So this was a 483 that was issued in June of 2015 to AB Science. So you can go and read the entire warning letter that if you want to. But one of the things was the finding was the failure to report serious adverse events according to protocol specified timeframes and a failure to perform protocol required laboratory tests. Now, the comments were the proposed corrective actions make no provisions for assessing the effectiveness of your monitoring practice. So that's why it is so important that it isn't that you just identify the issue and that you do whatever corrective action you need, but you need to document how you assured yourself that corrective action worked. So that's why those three are grouped together, and that's why it's so important for you to realize that you need to think about each of those steps as once you've identified an issue, and then you need to do that. Now, one of the things that we've done is to differentiate issues as major issues and minor issues, and we do that because not every issue needs to have a full root cause analysis. Sometimes it's very clear. So if it's a major issue, that is where we do the root cause analysis and corrective plans. So when you think about a root cause, so what you're seeing up here is the symptom, okay? And this could be, and then what's underneath is the underlying causes, or what made that system occur. So it would be the roots. So they're not always obvious, and you could have more than one root cause for a specific symptom or issue. So that's how this may help you to think about what we're talking about with root cause analysis. So what you need to think about for that issue, and it isn't necessarily just a site monitor, it could be the whole team. So is this a systematic issue? Is it occurring across sites? So one of the benefits of an issue management system is that each monitor can enter issues into the system, and then our central monitor can look at those issues across all the sites and see, are we seeing that as a consistent issue? Or is this an issue we're only seeing at one site? So is it something that is an isolated incident? So again, thinking about tolerance levels, is this an isolated incident or is this a pattern of behavior? So that allows you to, again, determine the scope of the issue or the extent. And does it involve a single site? Is it all the sites that have maybe have one monitor? Is it all the sites that are in a specific region? Those are the kinds of things that you want to think about when you're looking at that, because that's helping you understand the scope, to understand whether it's just something that maybe one person didn't understand, or maybe it's one site's training was unclear, or it could be that it was one region that went through a certain training program. Each of these will help you to better understand, what the root cause is. And as I said, there could be more than one root cause, but that you devise and implement a corrective action plan for each root cause identified. So we just separate them as issues so that we tie one root cause to one issue, and that allows us to be able to more finely determine whether those have been corrected. So you want that intervention to be tailored to the specific root cause. And what I mean by that is it's not always education. So one of the things that you often see is, oh, they're making these errors, it's education. Well, that may not be the reason. It could be resourcing. It could be the way the protocol was designed. It could be the way the questions were asked. So you really want to understand why something happened so that you can tailor your intervention based on the specific root cause. And then what is that behavior that you're going to use that tells you whether your intervention was effective? And then you need to remember that if it wasn't effective, you need to go back and redefine the CAP or the CAPA, corrective action plan, or sometimes it's corrective and preventative actions, either one. I think both of them to me mean this are similar, but the idea is you can't just if it didn't work the first time, you can't give up. You need to go back and follow it through till that issue is corrected. Remembering that what we certainly do is we focus that on the major issues. And those are the ones that all have to have a corrective action plan. So what you want to do in your corrective action plan is to state the problem or the weakness, including the root cause and the extent of the problem. So define the problem. What is happening? What is the effect? What is the reason it's happening? And how can it be fixed? So there's a number of different ways you can do that. You can set up we set up a specific form that asks these questions so that we can keep track of this, and it helps us to better enhance the way we do our training and design our systems. So sometimes what you might want to do is just make a list of what are the possible root causes. So if we go back to that study that we talked about, the graphs of the rating scales. Okay, let's go back to that one and think about that. So for that one, remember that the scale that went from 5 to 4 is showing us that at least for one subject at that site, there were problems with the rating. So it could be that there were different people doing the ratings on different days. Now, this, again, as part of the training and the processes for the study, they did include a plan for what to do if the rater wasn't going to be there. So if a rater wasn't going to be there on a certain day, then the person that was going to cover for them needed to rate the subject together on the day before that. So if you think back on that process, and remember, this is a key efficacy, then what we want to do is to collect the information as to who did the rating, initials or something, and have the ability to collect it, have multiple people enter their initials so that you know, did they follow the procedure that's in there and have a rating done by a new person together with the old person on the day before there was going to be a change. So this is something that is a testable hypothesis. It could be that the rater didn't rate the subject correctly. Oh, I'm sorry. Let me just walk through, go to both sides, because I think it's a little bit helpful. So let's just say it was that a different person doing the rating on different days. Then the corrective action could be emphasizing the importance of that transition rating on a previous day. And you could check that. So you could implement this training for this one as you need to. And then you could very easily say that I'm now going to check that by looking at the rater's initials if you have that in your CRF so that you can check that. The second thing is that the rater didn't rate the subject correctly. So you know that you go through rating training, but people make mistakes. So is this a one-off thing? Is this somebody that just needs to have some additional reminders about how you do your rating? And the nice thing is, instead of just saying you need a reminder, you can give them an example of this specific subject so they can think about it. And is the PI doing the ratings, or is someone else using the PI's ID? Or this could be whoever the rater is. And for this would be a significant site performance issue that may necessitate a for-cause visit, on-site visit. So if you find that this is what happened, and again, as the site monitor, you need to go in and you need to talk to the site and understand, hey, tell me about that rating on subject 0505. Who did that? What happened there? It looks kind of interesting. So who did the rating? Oh, that was Susie because Joe was gone. Again, that's how you find out this information. And that's why the site monitor's job is so important, because you have that relationship with the site. So you want to be able to understand that, because then it depends on what you're going to do based on what the potential root cause was. And then finally, there is a situation where you can have two lesions, and if they're rated together, then in fact you could see a situation where you could go from 5 to 4. And if that's the case, nothing's needed. So again, if once you understand, oh, that person had two lesions, so that's why we had to rate, you then go to the rating of the second lesion, and that's the reason that happened. No problem, no issue. So that's what we're talking about, is if once you understand, it will determine what your corrective action is going to be. Okay? All right. So because this is a lot of material, I'm just going to see if there's any questions. Nope, no questions. So if you have questions, send them to me, and we will include those. We'll go through those today too. So please let me know if you have questions, and we'll go through those at the end. Now, so we talked about stating the problem, understanding the root cause. Now we want to implement that intervention. And as I said, that needs to be customized to the specific root cause, and you need to document that. Okay, so if you go back to everything that you've learned from the first day you were a monitor in clinical research, you need is that you need to document what you did. So it's really important that you document that. And then the third step is to determine whether that issue has been corrected. So one of the things you want to think about when you're determining your root cause, you're setting up your plan, is how am I going to know if they did it right? And put that in so we know what to look at. Is it I'm going to look at the ratings for the next 5 subjects and make sure, or I'm going to identify where there's a transition and make sure that they've done the ratings together. It could be any number of things, but what you want to do is figure out, how are you going to know that the issue was corrected? Who's going to determine that that issue was corrected? What's the time frame for checking that? And then how are you going to document that that was corrected? Or if it's not corrected, what are you going to go back through and try to do again? So that's why this step of making sure that you know how you're going to make sure it's fixed is really, really important. Okay? So let's look at the example we have. So this was, go back to our rating one we talked about. The ratings occur on different days by different raters. So we knew that because we collected the initials of the people who did the rating. So what we did is met with the site, reviewed the transition process, and then the follow-up is that the ratings will be consistent for the next 5 subjects that have crossover between reviewers. So that is the way that we were going to go ahead and follow that up. And if in fact that's correct, we're good to go. And we can document that the site has implemented the correct transition process, and we've closed the issue. Now, one of the things in the ICH guideline, the new guidelines that came out from the ICH, is that any major issues, they think their recommendation is those be addressed from a data integrity standpoint in the clinical study report. So keep your eyes on that. That hasn't been adopted, but that may be the expectation in the future. Now, one of the things that we've been following, we follow RBM from all the different aspects, and Chesapeake IRB did a really nice webinar, and I've got the link here to it, and you can access it there. And in that, and then we've talked to them subsequently to the webinar, is that when a sponsor and a site, when they sign an agreement with an IRB or an ethics committee, it's the expectation that the sponsor or CRO will share any issue that affects study integrity and subject safety with the IRB. So that's always really difficult. So first of all, that's a change because that's the sponsor CRO, as opposed to in the past, everything that was considered an issue for subject safety was supposed to come in through the site. So that's one change. The second is, as we talked to them about implementing this, one of the ways that they recommended, and which we are working with now, is that if you have to implement a CAPA, then that should be reported to the IRB. So let's just say a study where the site has a number of kind of questionable issues with crossouts on informed consents, or the ink looks like it was the same person that signed the informed consent and did the and the writing looks like the same person signed the informed consent as well as witnessing it. Those kinds of things would require a CAPA, and those would be the expectation from Chesapeake, is that those would be the things that you would report to the IRB. So this is a changing area right now. But what I took from that webinar, and I thought it was a very good one, was that it is the IRB's responsibility to look at study integrity and subject safety, because their whole goal is to assure subject safety. So if a study is not being done as it was originally designed, then that changes the risk-benefit analysis for the subjects. And if there are major issues that are going on that are requiring a CAPA with your work with the site, then it's the responsibility of the sponsor, the CRO, or the site, but somebody to communicate that to the IRB. So we are starting to work under that assumption, because we think that, again, we're all interested in the same thing, and that is maintaining subject safety and having good data integrity and trial integrity. So I put this on here so that you guys would be able to go to this link and listen to this IRB as well. So I thought it was an excellent discussion of this. So in summary, here is what you need to do as part of the risk assessment and mitigation. Okay, you need to identify those potential risks. You need to quantify the risks. You need to assure that I didn't finish this one out, I'm sorry. So you need to assure that you know how you're going to look at the data, that the data is available, the reports are available, so that you can do the assessments that you need to. You need to determine the tolerance risks. So which are the sites that are going to, what's the level at which you are going to say, we need to do something, and which is the level that you can say, oh, this is noise. You want to monitor for the occurrence. You want to understand, why did that error occur or that issue occur? Figure out how you're going to do that remediation based on the reason for the occurrence. And finally, you're going to monitor for correction of the behavior. So these are the key points, and this is what, if you take home nothing else, but this is what you're going to do as far as risk assessment. And once you identify issues, this is what you're going to do to correct those. Then you will have really made great progress in implementing the first really critical part of risk-based monitoring. So as I said, let me see. I think that's the last one. I'm going to just see if anybody has any questions. I know that we have a small group here, but let's see. So, okay. Can we work with other colleagues on coming up with corrective actions and follow-up, or is it done with the team? So it kind of depends. If the issue is identified as being a specific issue at the site, then I think it's the monitor's responsibility to understand why it happened, but the corrective action plan and what you need to do, and whether it's training, of course, this is a team activity. So what you're doing now is you are we're changing the whole way that we do oversight from we're going to work as individual silos. The site monitor is going to do one thing, the data manager is going to do one thing, the medical monitor is going to do another, to having a team to really think about these things. Because as in the white paper that we've released, is that just even a specific thing like the percentage of screen failures is higher than what you would expect. So let's just say that's gone outside of the tolerance level. Well, that has different impact whether you're a project manager and you need to think about budget, and you need to think about timelines, and the medical monitor, it has a different impact. Does this mean that are there things that we've done as far as inclusion exclusion criteria that may be too tight? Is it from the monitor standpoint, does this site really understand the protocol? Is that why they're getting so many screen failures? So each person on the team really plays a role here. So if you've gotten to the point where you're implementing a CAPA, I absolutely think doing this as a team is a really good activity, especially as you're developing your confidence and everything else that has to do with that. So I think that's it's a great question and a good way for you to think about working on these as a team. If there's no other questions, then what I'm going to do is close. We knew this was going to be a short session because this is makeup, and we'll look forward to talking to those of you that are going to come tomorrow so that we can go through the rest of the information start the information around technology. And I know some of you may say, "Ooh, I don't like technology," but please, I think you're going to find it's really, really useful to you. And there's a lot there that can really help you do your job. So thanks, sir, very much for attending this makeup session. Oh, one other thing. From now on, if we need to do a makeup sessions, we're going to do those on Wednesday at 1 p.m. so that we're not going to poll people or do anything like that. It's too difficult. So if we don't get through all the materials, then what we'll do is we'll have makeup sessions on Wednesday at 1 so that you can do that. And if you can't attend those, we will make sure that those makeup sessions are posted on our YouTube channel so that you can get access. So thanks very much. Have a great day, and I'll see you tomorrow. Bye-bye.